Hello, everybody. I am going to go through the lecture that I am covering tomorrow during lecture so that you have a little bit earlier. I'm doing this so that you guys will have some time to kind of digest things. We are going to go through each one of these pieces. It just will be a little bit quicker. So hopefully this gives you a little bit of time to go through it, digest it, acclimate to the DNA piece because we want to be able to cover this, but also that cover some of the review for the test this week. The test starts on Tuesday, so I want to give you time to kind of go through it before we even get going. So we're going to get started right now on our lecture. Let's go here. Hopefully you can see that. All right, so I'm going to turn off the subtitles. So we are going to cover a couple of things. First, we're going to cover a case study or case studies that had to do with DNA replication or DNA connections that were connected to an assault and murder, death, that kind of thing. We're going to look at a brief review of DNA structure and then how DNA replication works. On the exam, you will see the DNA replication for sure, and then, of course, the DNA structure piece that you're going to need to be able to identify. All right, so we're first going to look at the case study of Kennedy Brewer. Kennedy Brewer was a man that had a girlfriend. Her name was Christine. She had a 3-year-old daughter, who was raped and murdered, and then they found her body about 3 days later in a river. And so, because of where he was, and it was just a boyfriend thing, they actually arrested Kennedy Brewer for the crime. And so he claimed to be innocent. There was no credible evidence that linked him to the murder, but he was convicted mainly on the testimony of a forensic dentist who used bite mark evidence to claim that Brewer was the child's murderer. So the sad part is that the defense introduced him as a licensed dentist and a founding member of the American Board of Forensics for orthodontia. I can't even say the word. Odonto teeth studies. He testified the marks were not human bites, but they were probably the result of insect bites that were on the body sustained from being left in water in the creek for over a period of time. And he said it would be impossible to leave repeated bite marks like that or impressions like that with only the two top teeth. But at the time of the trial, the dentist had already been discredited and suspended from the ABFO. And so he was charged and convicted of capital murder and sent to Mississippi's death row in 1995. And he waited in jail that entire time for the 3 years for the trial to even begin. So he was convicted, sent to prison, was going to be put to death. He was on death row. So when we're looking at DNA forensics analogies, there are a lot of different ways to do forensics analysis of DNA, and the most common in the US is the short tandem reanalysis, which is the original 13 loci tested. There are now 20, but these are the originals. And so the DNA from the semen collected at the crime scene was tested and compared to Kennedy Brewer's. And they didn't match. Um, at least that's what there was a match, but there wasn't. It wasn't, there were pieces that were not complete because of the 13 versus 20. So they were looking at the most common ones, which are usually a lot more common between people. And so those others that were missing would have been the exonerating piece. So remember in 2001, this is in 2001, so they had only used 13 out of the 20 now. And so when they looked at the DNA from the 13 to the 20 and placed it in the genome, and they used what we call a gel electrophoresis. So they copied it a lot of times, loaded into the DNA sample into the gel, and then they applied a current. And we all know that the reason that it works so well is because DNA has a charge. a negative charge. So it's attracted to the positive side. So they load the DNA samples on the negative side of the current, and then they run it to the positive side. And so the DNA basically runs through the gel, and it goes according to size, and it goes through polarity, and it goes through weight. And so it leaves this lovely little print on the analysis gel. And so it would look something like this. This is not from the case, but you have a crime scene, and then you would have suspect 1, 2, and 3. And if you look at the crime scene versus the suspects, the one in the middle, the one in yellow, is the one that actually would be the suspect, the crime scene person. And so that's kind of how it works. And so for Kennedy Brewer remaining in 5 years, after DNA showed he was in a sense pretty sad. So in 2001, the DNA evidence excluded Brewer. He was moved off death row but kept in prison. And then Mississippi wanted to try him again for the same case. And so they finally decided to contact the Innocent Project, and was able to get an investigation reopened in 2007. So this is again about 6 years after. And so he was able to be exonerated finally in 2008, and he spent 15 years in prison for a crime he did not commit. And so he actually received about $500,000 in compensation from the state of Mississippi, but because of his past and because he was in prison, he still to this day has a really hard time supporting himself even now. Another project, um, again, the crime scene turned out to match one of the original suspects. So then they look back at all of the other DNA samples that they had. And a guy named Justin Albert Johnson confessed to raping and murdering Christine Jackson. He also confessed to raping and murdering another 3-year-old. And so if they would have just actually done due diligence, they might have found this out a little bit earlier. So he spent 15 years in prison for something he didn't do. Another project, or another innocent project that they were looking at, was another case by LeVon Brooks, who was wrongly convicted in that case as well. And he had been in prison for 16 years. And then, of course, were able to secure his exoneration and release in 2008. But sadly, he died of cancer in 2018. So he was only out for about 10 years before he actually died. But he was exonerated from the same case as well. So the fundamental concept of this is that although we as humans have a sequence of genes that are usually the same, there is variation in the DNA sequence between individuals. And so each person has a unique genome. And so in order to prove an innocence, they had to actually look at the entire thing. And again, that time frame where that project was going on, it was still pretty early in how to use DNA to declare innocence, or to show who the suspect was. It was still pretty early. So the central dogma, you're going to see this over and over again, DNA to mRNA proteins, but there is these pieces that go along with it. So DNA has DNA replication. When we go from DNA to mRNA, there's transcription. That's what's occurring there. That's what happens. That's the process. Translation happens between mRNA to proteins. And then, of course, that's what we get at the end, is that piece. So always remember that that is the process. You'll see this again and again and again. It will show up on tests, just kind of pop up there. So make sure you realize that here, DNA, we're replicating it. If we're going from DNA to mRNA, transcription is happening. If we're going from mRNA to proteins, it is now translation. So those are the processes you want to keep track of. This is a snapshot of the properties of life. So storing, exchanging, using information, all of this goes back to the idea that basically the properties of life that are needed in order to show something's living. So self-replication of DNA, that a life form will evolve. All of these fit into those, DNA fits into those three categories. So we have the genome, the transcription, the translation. the dogma, the central dogma that we discuss, how DNA can replicate itself and use that replication during cell division. Then, of course, we're looking at mutations, the source of all variations, mistaking stakes in DNA replication that causes those mutations, and again, evolving in the process. So we're going to look at a couple of things first. So I want you to look at this and tell me, and you can even pause it here if you're not sure. You can look and see or tell me, is this a monomer of DNA or RNA? And if you're looking at it, I'm going to give you a second. And you can pause it right now and look at it and try and figure it out. Okay, if you paused it, um, take a look at the structure already. Hopefully you already recognize that it is a DNA nucleotide, which is the monomer for DNA. So you have your phosphate, you have your sugar, and you have your nitrogenous base. So what they're asking is, how can you tell between something that's DNA and RNA? Well, RNA has a different structure than as far as what's included into that nucleotide versus DNA. DNA is deoxyribose, which means it's been deoxified. So if you look down here at the bottom where it has the hydroxyls, if this were DNA, this would be gone. That oxygen would be gone. So this is definitely RNA because we have two hydroxyls on those corners. So if we're looking at each part of the nucleotide, we have the phosphate group, we have the pentose sugar, and we have the nitrogenous bases. So make sure that you can name or look at that and do those names itself. When we're looking at the nitrogenous base, notice that it is, this one actually is either going to be, its structure will tell you kind of which base that is, and we'll get to that in just one second. But definitely RNA, right? Because it has the two hydroxyls. Again, if it had a missing oxygen, then it would be DNA. So I want you to go through and answer this question for me. Which is the 3 carbons? So this is now we're counting carbons on the sugar. So we're looking at this structure right here. So you can pause me for a second if you want and see if you can find the 3 carbon sugar. Which one is it? Okay. All right, hopefully you figured out that first of all, this is our 5 carbon right here. So we're going to go backwards to the nitrogenous base, and this is easy because it's set up nice where it's got the phosphate on one side and then the nitrogenous base is on the middle. So we have 5, 4, 3, 2, and 1. There's our carbon. So again, this is 5 carbon, 4, this is our 3rd carbon, 2, and 1. So the 1 is connected back to the nitrogenous base, where our 5th carbon is always going to be connecting into that phosphate. So hopefully you have that all in your head. So we're going to give a little bit of background on DNA structure and how that works. Watson and Crick, the most famous 2 people, both of them, they won a Nobel Peace Prize for this, decided or was given the credit of the double helix structure in 1953. So they actually built a model of the DNA structure, and it took them quite a few years. This was right after the war, right? And the sad part about this is that they used other people's information. So they came up with the structure, but they were stealing information from other universities at the time and using other people's research to do their own so that they could get credit for the double structure. So one of the people that they took information from was Rosalind Franklin. So she was an X-ray crystallographer of the DNA model at King's College, which was right there in London. And Watson and Crick had friends that worked in her research department. Her own boss was one of their friends. And during this time, there was this huge big race to figure out what DNA was. And the American, Watson and Crick, both of them were trying to figure it out before someone else did. And so they knew after watching some of her lectures that she was onto it. But back then they didn't think women were very smart and intelligent. And so they thought, well, she doesn't know what she has. So they had her boss actually got this photo, which we call Photo 51, stole it from her office, took it from her office, and sent it over to Watson and Crick. And once they saw that, they used her information that she had researched to create the DNA double helix. They also used another guy's information regarding the combination of which ones match to each other. So cytosine versus guanine, adenine to thymine. They used that research from another scientist at another university to come up with that plan. And so they won the Nobel Peace Prize. And then Watson, which that book is still actually, you can get it online. It's an interesting read. He basically like tore Franklin apart. And the sad part is that she actually went on, she left King's College, went on to another research facility, worked with viruses, became very famous for her work there. And her virus team that she worked with at the university gave her, they actually won a Nobel Peace Prize as well from the research that she did with them. But she actually died not too long after this all happened, 1958, I think, of cancer from her work using the X-ray machines because they were not well done. And so she actually died before both of those groups got the Nobel Peace Prize. And the sad part is Watson and Crick did not give her any type of credit, but her virus group did give her credit for their work. So she was named posthumous, but she did not receive the award. And it's funny how her work was involved in two groups that actually won the Nobel Peace Prize. All right, there's your history. So here's the paper that described the pairing. So this was not even Watson and Crick's work. They published it later. So they had three different types of publishing pieces in that newspaper. They published um, their work. They published the guy who did the base pairing, and then they published Rosalind Franklin's paper last. And then they said that they came up with her idea, or they basically came up with the plan, and that Rosalind Franklin actually is the one that just solidified their ideas, and that she agreed with them, not that she actually was the one that actually had the information. So this base pairing is what happened with all of that history. A to T, C to G. The way that I remember this is that if I say apples in the tree, car in the garage, it's easy to remember that those two fit together. Another couple of things that go along with this base pairing is the structure. So if you look at adenine and guanine, they actually have a two-ring structure. So right there, right there. This is what makes them purines. If it's two rings, it's got a purine. And then pyrimidines, thymine, and cytosine have a one-ring structure. And so they're pyrimidines. So if we're looking at also the structure within, when we're talking about A to T, there are two hydrogen bonds versus C to G, which is three. So again, these are hydrogen bonds, which makes it nice for DNA because it's not a very strong bond, but it's a good enough bond that keeps it together. And it would require a ton of energy for that to actually break apart and be able to be used. And so the hydrogen bond is a bonus for these two, and it allows again, the DNA to break apart fairly easy and not have to use a ton of energy to actually do replication or to do transcription. Another thing to note is that there is polarity in a DNA strand. So if the polarity is indicated by the 5 to 3. So when we're talking about how it's running, this is basically what we mean. So DNA runs from 5 to 3, which means that we're coming off of the phosphate here, and the next one down, whoops, sorry. The next one that you're going to see if we're connecting another nucleotide is that 5 carbon sugar. So if you move this guy this direction, here's that 5, and then we're connecting off of the 3 to the next one. So the top of the phosphate, or the bottom of the phosphate that's connecting into a new nucleotide. So a phosphate to the 5 carbon sugar is at the 5 carbon mark, and it's connecting to the next nucleotide on the 3. So this is running on the 5 end to the 3 end. So it's ending on the 3 end here. So on this point, we're looking at the opposite. It runs opposites coming in on the 3 end. So if we count these, 5, 4, 3, 2, and 1. So this phosphate is connected to the fifth carbon. This phosphate is connected to the fifth carbon. Here we go, 5, 4, 3, 2, 1. Again, 5 to 3 ends, that's how they run, and that's the polarity. So anytime we do replication of DNA, we have to go off of it has to be 5 to 3 in order for it to work when we're sending it out for mRNA. So if we're looking at the in-class work, I'm going to let you guys try and do some of this. I will verify it tomorrow. So for a second, I want you to label the carbons 1 to 5. So what does that look like? Making sure that you can find it. Label where the phosphate group is attached to this nucleotide. So looking at this one right here, where would the phosphate group be? Would it be here or would it be here? And then label where the base is attached. So the nitrogenous base, whether that's cytosine, guanine, whatever it might be. And then label where the next nucleotide is going to be attached during DNA replication. So go ahead and do that now. So I'll pause for a second. And then again, tomorrow in class, we're going to go through this part for sure so that you guys have a clear understanding of how this actually works and where the connections are. And I'm going to give you a hint right now. This is flipped. It's inverted. So take that as you want. And then tomorrow we'll go through it a little bit more. Okay, so hopefully that you have that part done. We're going to look at DNA replication. So here's one strand of DNA. We're going to write the other strand that matches it, including polarity. So polarity is indicated by the 3 to 5. So if I'm writing this, we're going from, we're going the opposite direction. So instead of being 3 here, it's going to be 5. Good. Hopefully you said that. And then remember when we're matching these up, it's apple in the tree, car in the garage. Car in the garage can be garage in the car. However, it's backwards. It runs both ways. And then A to T, which is apple in the tree, runs both ways. So hopefully you're doing this. So pause it for a second so you can write down what you think it is. And then again, tomorrow we'll do this in class. So pause, write, go. Okay, hopefully you pause that and this is what you came up with. So you have the opposite run. So from 3 to 5 here, we're going from 5 to 3. Car in the garage, apple in the tree, apple in the tree, apple in the tree, car in the garage. Do you see how it works? So this 5 to 3 is the polarity. That's what that means. It's including the polarity is that 5 to 3. So if this was 5 to 3 and I asked you to write the other DNA strand, the polarity on the other DNA strand would go back to the 3 and then it would end in the 5. So that's what it means by polarity. All right, when we're looking at DNA as a whole, we have DNA, we have just the normal strands like you saw, just the baby ones. So this is how it starts until we start looking at a gene segment. So a gene segment would be a ton of DNA. Maybe that's eye color in the gene. DNA itself is the entire swath of DNA until you get to the chromosome. And then we're looking at a very condensed element of DNA, and they use that for mitosis and meiosis in order for it to be able to divide rather easily. So when we're looking at DNA, it's this baby guy. Genes are like a bunch of the DNA together, and then in order to get a chromosome, we're looking at that very condensed piece, and then the genomes themselves is the entire look of someone's DNA as you're going through. So one of the research pieces, Melton and Stahl experiment, was looking at how things actually replicate, how DNA models, which way they replicate. So they designed this test in order to determine 3 possible models of DNA replication. And so they were looking at conservative, they were looking at semi-conservative and dispersive. So we have a parent DNA molecule, and they wanted to figure out how 2 DNA molecules came out of the parent. And so they use what we call the DNA of the E. coli. They grew some E. coli for several generations, and then they transferred the E. coli to the N4, which is a very heavy nitrogen. And then they used the sample after 1 or 2 rounds of cell division, and then they figured out how it actually was being replicated. And so This experiment, again, they used the heavy nitrogen for several generations. They used the most common isotope of nitrogen, and then they allowed those E. coli cells to divide and track cell divisions at the same time. And then they extracted the DNA at a certain time point and then looked at it, put it in a centrifuge, and then looked at the density to see how it was working. So they determined that DNA replicates semi-conservative. So basically the double helix separates and then each strand is copied. The new DNA molecule has one of the old strands and one of the new strands. And so this is basically broken apart and you have a new strand in green, a new strand in green, and the old strand in the blue. So here's how it actually happens. These are the pieces of how it actually replicates. And you're going to want to take good notes on this because this is in the test. It is on your sheet, a worksheet. We're going to go over it tomorrow, but it is on the test. You've got to be able to determine what each one of these do. So the helicase, which is an enzyme, and it basically goes in and basically unwinds DNA to produce that replication fork. So here we're running 3 to 5 on the top, 5 to 3 here on the bottom. So again, helicase is the one that does that. Then a primase comes in and makes an RNA primer. So basically it's telling the next step that RNA, the DNA polymerase, where to start replicating. So the primase connects in, here's where you're going to start replicating, and then the DNA polymerase comes in and makes the new strand of DNA. And it only synthesizes from 5 to 3. So we're going back this direction. So this is 3, so we're doing the new DNA from the 5 to 3 direction. So it sometimes when we're looking at DNA polymerase, it's just building. It does some proofreading. Sometimes it adds in the correct bases but then it goes back and proofreads and then it removes and replaces it, but not always. It does try to do that, but now that's where we're getting into mutations. So mutations is that permanent change in the DNA. And then, of course, this leads to genetic variation. So when we're looking at mutations themselves, they can be spontaneous or induced. So if we're looking at spontaneous, this is basically just it's happening on its own. It's naturally happening. No environmental pieces are causing it. But if it's induced, it means that there's an environmental cause. And you can think of several different ones while you're doing this, like for instance, smoking or exposure to anything that's like a chemical. So those environmental pieces that cause mutations in DNA. And then again, most of them are due to DNA replication errors. And this again takes us back to the result. These mutations cause genetic variations, which we want. A couple of other different things is that there's a leading strand versus a lagging strand. And when we're looking at both of those, it's just how they're actually replicated within the process of replication. So the leading strand is going from the 3 to 5. It's continuously being done. It doesn't stop. But when we're looking at the lagging strand, this is discontinuous. They do sections and then they add in different elements to make it complete. And so again, the leading strand is just continuous replication. This one, it kind of goes backwards and then they're filling in with different pieces. And so the DNA removes any of the RNA plate pieces that are being held in there and then it replaces it with DNA. And so now we have a lovely strand of DNA with that DNA polymerase that's removing the RNA and replacing it. Once we have that completely finished, we now have the ligase, which actually comes in and seals all of these pieces of DNA. So now we have one solid strand of DNA, and the whole thing is complete. So when we're looking at different types of other ligase, DNA ligase, again, is something that goes in and basically clicks everything together. So Baltimore Oliveira, he actually is a professor at the University of Utah. He's been there since 1970, and he discovered and characterized a E. coli DNA ligase, which is a key enzyme in genetic engineering when we're looking at DNA technology. So he discovered that there were some small snail toxins most of which are some small peptides that bind to an ion channel and those receptors in the nervous system, and it actually helps with pain medication. So they're using this type of ligase, which is helping with this particular pain medication. It's funny because again, um, it was discovered in his lab um, from another student. So he runs the lab and he had people working for him underneath that. And the student that actually discovered this was a student that actually graduated from high school, had just graduated from high school. So his discovery actually helps and is now an approved pain medication. When we're looking at prokaryotic replication, there's two different ways to do this. This is the theta type replication. It's not the only mechanism for prokaryotic replication, but it is used, and most of the time we're using a uh, bidirectional replication. Usually that's what that is. If we're looking at eukaryotic replication, we have multiple linear chromosomes, so we're using that replication fork per chromosome. Um, so hopefully Hopefully that helps. Hopefully you'll have time to review this by tomorrow. If not, we're just going to go through it rather quickly so we can get to the review. Hopefully that made a little bit more sense and that you are a little bit more prepared for lecture tomorrow. Again, I'm going to go over all of this tomorrow. I just wanted you to have a little bit of a uh, pure upfront load, a frontal load of this information, and hopefully this will make sense or a little bit more sense tomorrow in class. Again, I will see you guys tomorrow, and have a great weekend, and we'll see you on Monday.